In this chapter, we're going to study the sequence of events from fertilization of a secondary oocyte by a sperm cell to the formation of a newborn baby. We'll focus on the developmental sequence from fertilization through implantation. Next, we'll look at embryonic and fetal development. Then we'll look at labor, and finally give birth to a baby. We'll also examine the principles of inheritance, which is simply the passage of hereditary traits from one generation to the next. So sexual reproduction is the process where organisms produce offspring by making sex cells called gametes. Male gametes are called sperm cells, and female gametes are called secondary oocytes. These cells are created in structures called gonads. In the male, the gonads are the testes. In the female, the gonads are the ovaries. Yes, females have gonads. Gonads are simply sex cell producing structures. So fertilization initiates a whole cascade of developmental events that, when they're completed properly, is going to give birth, or going to lead to, a healthy newborn baby. So pregnancy begins with fertilization and then proceeds to a period known as the embryonic period, and then fetal development, and again, ideally, about 38 weeks later, or 40 weeks after the mother's last menstrual period, a baby is born. So this period of prenatal development, or the development before birth, is the time from fertilization and includes both the embryonic and fetal periods. The prenatal development is divided into 3 calendar months, and we call those trimesters. During the first trimester, the most critical stage of development, by the way, all of the major organ systems begin to form. And because of the extensive and widespread activity that goes on during this time, it's also the period when the developing fetus, or the developing embryo, is most vulnerable to the effects of drugs and radiation. So the early part of development is the embryonic development, and then we'll talk about when fetal development happens. And all of this is during that first trimester. So anything that goes wrong during that first trimester, when all the organ systems are forming, can have very substantial effects on development. The second trimester is characterized by the nearly complete development of the organ system. So by the end of this stage, the fetus has very human-looking features and pretty well formed organ systems. The third trimester represents a period of rapid fetal development, and the fetus pretty much doubles its weight. During the early stages of this period, most of the organ systems become fully functional. So the embryonic period extends from fertilization through the end of the 8th week of development. The first week of development is characterized by several very significant events that include fertilization, cleavage of the zygote, the blastocyst formation, and implantation. So we'll begin by examining fertilization. So during fertilization, genetic material from sperm cells and egg cells are going to merge. Now the sperm cells have one copy of all of the chromosomes, so we call those haploid sperm cells, and the egg cell has one copy of all the chromosomes. We call that a haploid egg cell, and when their nuclei get together, they create a diploid cell. So that means now there are 2 copies of all the chromosomes. So the about 200 million sperm that actually enter the vaginal canal during an ejaculation, fewer than 2 million actually reach the cervix of the uterus, and only about 200 actually reach the secondary oocyte, which is out in the fallopian tube. So fertilization normally occurs in the fallopian tube within 12 to 24 hours after ovulation, or the release of the egg cell. Sperm cells, though, can remain viable after ejaculation for about 48 hours once they're deposited into the vagina. But again, the secondary oocyte is only viable for about 24 hours. So pregnancy is most likely to occur if intercourse takes place during a 3-day window. From 2 days before ovulation, because remember the sperm cells can last for up to 48 hours, to 1 day after ovulation, because that egg cell is only viable for about 24 hours. So sperm swim from the vagina into the cervical canal by whip-like movements of their tails that we know are called flagella. And the passage of the sperm through the rest of the uterus and then into the uterine tube results mainly from contraction of the walls of the uterus and the fallopian tubes. Prostaglandins that are a part of the ejaculation are believed to stimulate uterine motility at the time of intercourse into the uterine tube. So the contractions of the uterus are going to help to move those sperm cells throughout the uterus and then out into the fallopian tubes. So sperm cells that reach the vicinity of the oocyte within a few minutes of ejaculation are usually not even capable of fertilizing the egg until about 7 hours later, because they have to undergo a process known as capacitation. So that's just a series of functional changes that cause the sperm's tail to beat even more vigorously and prepare the plasma membrane of the sperm cell to fuse with the oocyte's plasma membrane. So during capacitation, the sperm are acted on by secretions of the female reproductive tract that result in the removal of cholesterol. Remember, cholesterol gives stiffness to membranes, glycoproteins, and proteins from the plasma membrane that surround the head of the sperm cell. So only those cells that have been capacitated are capable of being attracted by and responding to the chemical factors that are produced by that oocyte. A lot going on there, right? So for fertilization to occur, a sperm cell first has to penetrate 2 layers. The corona radiata, which is made up of the granulosa cells that surround the secondary oocyte, and the zona pellucida, which is a clear glycoprotein layer between the corona radiata and the oocyte's plasma membrane. So the acrosome, which is sort of like just a little helmet that covers the head of the sperm, contains digestive enzymes, and these acrosomal enzymes and the strong movements of the sperm cell tail help it to penetrate the cells of the corona radiata and then come in contact with the zona pellucida. On the zona pellucida there are receptors. Those receptors cause an action to occur in the sperm cell. Once the sperm cell binds those receptors, it triggers a reaction called the acrosomal reaction, and that causes the release of the contents of the acrosome. And remember, it's all those acrosomal enzymes in there. So those enzymes then digest through the zona pellucida, and the lashing of the sperm cell tail pushes that little sperm cell on forward. So even though many of the sperm cells may bind to those receptors and empty out their contents of those enzymes, it's only the first sperm cell that penetrates the entire zona pellucida and reaches the plasma membrane or cell membrane of that oocyte that's going to get in. So a fusion of the sperm cell with the secondary oocyte sets in motion a series of events that blocks an action called polyspermy, which is fertilization by more than one sperm cell. So within just a few seconds, the cell membrane of the oocyte depolarizes, and that triggers the release of a lot of calcium ions inside the cell. That then is going to stimulate exocytosis of these secretory vesicles from the oocyte, and the molecules that are released by this exocytosis inactivate the sperm cell receptors, right? So the sperm cells can't bind to the zona pellucida anymore, and it also causes the zona pellucida to harden. So no more sperm cells can get in. So once a sperm cell enters the secondary oocyte has to complete meiosis 1. Remember when we were talking about making sperm and egg cells, and I mentioned that during meiosis, the only time that meiosis 2 completes in the female is if fertilization occurs. So yes, now fertilization has occurred, so the oocyte can complete meiosis 2. So first thing that happens is it divides into a mature egg, a large ovum. Okay, and then a second smaller polar body is created, and it's going to fragment and disintegrate. So the nucleus of the head of the sperm is going to develop into a structure called the male pronucleus, and the nucleus of the fertilized egg cell develops into the female pronucleus. And after these two pronuclei form, they fuse together and form a single diploid nucleus. So now there's two copies of all of those chromosomes. So the fusion of the haploid 2 pronuclei restores that full number of chromosomes. So now we have a diploid cell called a zygote that contains all 46 chromosomes. All right, so let's talk just a minute about some of the things that can happen once fertilization has occurred, and this is something that I think is really cool, and that's the whole process of twins. So dizygotic or fraternal twins are produced when you have two secondary oocytes fertilized by different sperm cells. So these zygotes and these children, once they're born, are the same age and in the uterus at the same time, but genetically, they're no more alike than you and any of your other siblings. Dizygotic, two zygotes form. So digozotic twins may be both male, both female, or you may have one of each. They're not genetically identical. But monozygotic twins, those are the ones we call identical, develop from a single fertilized ovum. So they contain exactly the same genetic material because one sperm cell fertilized one egg cell. So these are always going to be the same sex. They're both female or they're both male. Monozygotic twins arise from the separation of the developing cells into embryos, and that's about 99% of the cases, and it occurs before about 8 days during the developmental process. So when separation doesn't happen early in the process, usually after 8 days, it's more likely to produce conjoined twins. So this is a situation where the twins are joined together and share some of the body structures. Now this may be that they simply share an arm, or it could be that they share a brain and some organs. So it just depends on the particular time that the separation did not occur. After fertilization, a rapid mitotic cell division of the zygote begins, and that process is called cleavage. The first division of the zygote begins about 24 hours after fertilization and is completed about 6 hours later. Each successive division after that takes slightly less time. By the end of the second day after fertilization, the second cleavage is completed and there are 4 cells. By the end of the third day, there's 16 cells. Successive cleavages eventually produce a solid ball of cells that we call a morula. Morula means mulberry, and that's sort of what it looks like, just a ball of cells. The morula is still surrounded by the zona pellucida and is about the same size as the original zygote. By the end of the 4th day, the number of cells in the morula increases as it continues to move through the uterine tube toward the uterine cavity. Remember, fertilization takes place in the uterine or fallopian tube. So the morula enters the uterine cavity on about day 4 or 5 and starts to reorganize and forms a blastocyst cavity. So once the blastocyst cavity is formed, the developing mass is called a blastocyst. Now it has hundreds of cells, but the blastocyst is still about the same size as the original zygote. During the formation of the blastocyst, 2 very distinct populations of cells arise. The inner cell mass is located internally and will eventually develop into the embryo, and the trophoblast is the outer ring of cells that surrounds the sphere and forms the walls of the blastocyst. It's going to develop into the outer chorionic sac that surrounds the fetus and then will eventually develop into part of the placenta. The placenta, of course, is where nutrients are exchanged from mom to baby and waste products from baby to mom so mom can get rid of them. So around the 5th day after fertilization, the blastocyst hatches, I guess you could say, from the zona pellucida by digesting a hole in it with enzymes and then squeezing through the hole. So the shedding of the zona pellucida is necessary in order for the next step, which is implantation, to happen in the endometrial lining of the uterus.